Hi everyone, welcome back to the channel. This is Dr. Hasi. Today's topic is bio bsc1010 lab test 1 study guide. So we are going to cover 3 chapters or 3 labs in this video that will be tested on lab test 1. Okay, please always remember to finish up your McGraw-Hill Connect labs and make sure that you review all the labs so far, which are metric system, microscope, cell, and chemistry of life or food tests, okay, or macromolecules. So, and also double check your syllabus, your lecture lab notes, and make sure that you review them for just making them, refreshing them. Okay, so lab 1, metric system and quantification of data. So a few key terms you need to be familiar with: metric system versus English system. What are the differences? And we make sure that you will be able to convert from one to another. Also, like volume, weight, temperature. And again, same idea. The conversions, data, continuous versus binomial or discrete data, right? So data can be qualitative. That's like there is no numbers about that. It's more qualitative data, or quantitative, which we are more interested. Could be discrete or binomial, like color, eye color, gendered, number of eggs in a basket, stuff like that. That will be like basically you see here a bar graph, which is basically showing just numbers, whole numbers, right? Like 5x, okay, number of Facebook likes, eye color. But another type of data is continuous data. So like water temperature or drill depth in the ocean, grades in a class, right? As you see, this is a more a normal distribution type of data. That's we call continuous data, okay? So you can fit as many numbers here. They don't have to be whole numbers like binomial, like here, okay? Yes, no, or yellow, or blue, and stuff like, or pink colors, etc. So make sure that you know the differences between these, and also make sure that you basically know how to convert the temperatures from Fahrenheit to degree Celsius or Kelvin. And there is a formula for specific gravity, which is specific gravity equals weight of the substance divided by volume of the substance. If you have a 3-gram substance like this here, if you want to find out its specific gravity, you can use this graduated cylinder, put in maybe 5 milliliter water. If you put this inside here, and the difference in the volume here is gonna be its volume, like 1 milliliter. You see, it went up. Then 3 gram divided by 1 milliliter, which equals 3. Why there is no unit? Because actually 1 milliliter water is also about approximately about 1 gram of water. And the grams actually cancel out, and we have no unit for specific gravity. That's chapter 1. Lab number 2 is microscope and cell. Make sure you know microscope parts, the names, the total magnification, wet mount, right? You see here the compound microscopes, electron microscope, just the difference and which organelles we need to see electron microscopes. Remember, smaller organelles like ribosomes definitely require something like electron microscope. Objectives important, and they are actually, they have numbers 4x, 10x, 40x, starting with scanning, low, high, right? And then cell types, right? Bacteria, animal cell, plant cell. What are the similarities? What are the differences between these cells? So to make sure that you know your organelles, and then you can recognize a potato cell and an onion cell, or human cheek cells and bacteria cell. Lab 3 is chemistry of life or food test. In this lab, we tested 5 different food types in basically using different assays. Like for monosaccharides, we use Benedict solution, and then the positive color should be orange color. For polysaccharides like starch, we use iodine solution. Positive color change for this assay is black. For lipids, we use petroleum ether, and then we use brown paper. No color change, but the grease or no grease on the paper. And also for proteins, we use red test, and then the color changes again, purplish color. DNA or nucleic acid, very same, but the test name is ditch solution. We also looked at the pH. Remember the pH scale, 0 to 14, and pH can be adjusted. Actually, pH meter can be calibrated using the buffer solutions, buffer something that holds the pH in a specific location. So you can actually calibrate it, or you could use a pH paper. Basically, look at the color changes on the pH paper. More red, pinkish colors are lower acidic pHs, and then more bluish colors are higher pHs or basic pHs. So that's for all now. Thanks for watching. Just to recap, this is Lab 1 test study guide for bsc1010 Lab, General Bio 1 Lab. Again, thanks for watching, and please feel free to comment and subscribe to the channel, and best of luck with your studies. See you next time.